Methods for joining DNA fragments. Mainly three methods are used to join DNA fragments from different sources. Mainly it will depend that to which restriction enzyme the DNA has been fragmented or digested. जो पहला मेथड है ये बेस करता है on the DNA ligase जो कि isolate किया गया है E. coli से. So this DNA ligase from E. coli, it will mainly target those DNA fragments that have been digested with those restriction enzymes that produce cohesive or sticky ends. So it will mediate the formation of phosphodiester bonds between the DNA fragments that are having the cohesive end. Another method, it is also depends on DNA ligase, but the source is different. This DNA ligase, it is encoded by phage T4 that can infect the E. coli. Now this T4 DNA ligase, it will join or ligate the DNA fragments that are having the blunt ends. For instance, if the target DNA, it is digested with T3 restriction enzyme, the DNA fragments that will be produced having the blunt ends. So the ligation of blunt ends, it is mediated by T4 DNA ligase. The third method, it depends upon the ability of terminal deoxynucleotidyl transferase, and commonly it is also called as terminal transferase. This enzyme, it introduces homopolymer tails at 3' ends of the DNA fragment. Homopolymer tailing means that the tails contain a single type of residues at 3' ends. For example, either adenine, guanine, cytosine or thymine. How it is possible? We are going to discuss after some time. But now we are going to focus on DNA ligase. As I mentioned that DNA ligase, it mediates the formation of phosphodiester bond between two adjacent nucleotides. So it introduces bond between 5' phosphate and 3' hydroxyl group, because when two DNA molecules having the sticky end, they will join because of complementary base pairing. So gaps remains that may be 1 or 2 nucleotide base pairs apart. So this gap will be bridged and bond will be formed by DNA ligase. So in both cases, either it is E. coli DNA ligase or DNA ligase from T4 phages, so both will catalyze the formation of phosphodiester bond. But during their activity, their cofactor requirements may be different. For example, T4 DNA ligase, it requires ATP, जबकि जो बैक्टीरियम DNA ligase है, इसका जो कोफैक्टर है वो NAD+ है. NAD+ means that nicotine amide adenine dinucleotide. So both will split or hydrolyze after interacting with the DNA ligase, and AMP and enzyme complex will be formed, and this AMP enzyme complex will be used to form phosphodiester bonds. So this diagram, it indicates that how cohesive or sticky ends they can anneal and finally ligate. So here this DNA, it is basically digested with EcoR1, so the sticky ends, they are having single stranded ends that are TTAA or AATT. So they can anneal here, and the gap that is produced here, it is mainly breached by the activity of DNA ligase. So there is another diagram that indicating the action of T4 DNA ligase and the DNA ligase isolated from E. coli. So their cofactor requirements are different, like ATP for T4 DNA ligase and NAD+ for DNA ligase from E. coli. So both will hydrolyze and enzyme AMP complex will be formed here. So this is the target site, 5' phosphate and 3' OH group that needs to be breached or ligate so that phosphodiester bond will be formed. So enzyme will bind here and finally it will introduce phosphodiester bond within the DNA molecule.